We are going to talk about food supplements beyond our hype and what clinical science really tells about them. For this, and without further ado, I would like to welcome our distinguished speaker for today, Dr. Hamadullah. Please, thank you very much, sir, for joining us today. And it is now my honor to introduce you. As dear all, Dr. Hamadullah is an assistant professor at the School of Pharmacy, University of Management and Technology, Lahore. He holds a PhD in nutraceuticals and functional foods, along with postdoc fellowship and habilitation in food chemistry from University of Naples, Italy. His research expertise lie in nutraceuticals, functional food ingredients, and the sustainable valorization of the agri-food byproducts. And he has authored over 80 peer-reviewed publications, contributed to books and book chapters, and served as an editorial board of leading international journals. He is also the member of prestigious Society of Chemistry, American Chemistry Society, and the Phytochemical Society of Europe. Among his various accolades, he has received the Pride of Pakistan Award 2025 and the Early Career Medal by the Royal Society of Chemistry, which makes him a scientific milestone for the Pakistan scientific community. We are very privileged to have you, sir, and please guide us today through an evidence-based perspective on the food supplements. And now, without further ado, I would like to invite Dr. Hamad to start the session today. But before that, I would request that you all keep the mics muted so that the session is not interrupted. And thank you very much. Over to you, sir.
Assalamu alaikum and good afternoon, everyone. It is an absolute pleasure to welcome you to today's session on nutraceuticals beyond hype, what clinical science really tells us. First of all, let me also tell you that actually I am traveling, so I would like to keep my camera off to avoid any interruption in connectivity. The slide is visible.
Yes, sir, it's clearly visible.
Okay, thank you. Today, I hope to share insights from clinical science and translational research to help all of us distinguish between evidence-based benefits and overhyped claims in food supplements, nutraceuticals, and functional foods. So let me start. With some basic terminologies in this area. But before starting, we see two pioneers on this slide who have shaped the concept of nutraceuticals and food supplements, in other words, natural products. On the left side, you can see Hippocrates, one of the most famous personality in food science and natural products, often called the father of medicine, who said, let food be the medicine and medicine be the food. His wisdom reminds us that diet has always been critical to human health. On the right side, maybe many of you are not aware of this personality, Dr. Stephen Defeles, who coined the term nutraceuticals in 1989. Before Dr. Stephen or before 1989, people were working with nutraceuticals, but there was not a proper terminology like nutraceuticals. They just called it supplements. What Dr. Stephen did, Dr. Stephen coined the term nutraceutical that laid the foundation for modern research into foods with medicinal or health promoting properties. If I am talking about today, we build on both of their legacies by applying rigorous clinical science to evaluate these claims or claims of food derived products. Before we dive in, let's clarify the major terminologies. What are nutraceuticals, food supplements, and functional foods? And these are same, or there are big differences. Nutraceuticals are basically foods or parts of foods that provide medical or health benefits, including disease prevention. So this is very simple for nutraceutical. Nutraceutical is basically combining of two different terminologies, nutrition and pharmaceutical. So applying the concept of pharmaceutical to nutrition or nutrition derived factors, we call it nutraceutical. So it is a bit simple. Under the umbrella of nutraceuticals, two terminologies falls: food supplement and functional foods. So what are food supplements? These are substances that are added to the diet, but often formulated as pharmaceutical formulation to treat or prevent a deficiency. While the functional foods are designed to contain bioactive compounds with potential health improving or disease preventing value. So if someone of you go to pharmacy and he see on the pharmacy shelf multivitamins, for example, are the supplements containing the pharmaceutical product containing minerals, containing probiotics, all these are food supplements. But when you go to food market, and there you can see some of the shakes or milks that contain dietary fibers, that contain vitamin D, that may contain vitamin C. So these are basically functional foods. In functional foods, we use the food, but we add some of food derived by active components to increase functionality or functional properties of that food. So now let's come towards the main theme of this lecture: popular claims versus scientific reality. With the nutraceutical claims are everywhere, and it is one of the major issue of misusing or misleading nutraceuticals and food supplement for different health conditions. In the global market, nutraceuticals and supplements exceeds about $200 billion annually, if I am talking for the last year. With such widespread use, claims like improved digestion, control blood sugar, control blood cholesterol, or boosting immunity. These are basically the claims that are leading towards increased use of nutraceuticals. But are they effective for these conditions? Not always. Why? Because if we look into science, most of these claims are based on in vitro studies, animal studies, or human studies, but short-term uncontrolled observations. When we come to rigorous clinical trials, what we observe, because my research group has rich experience in testing different food supplement ingredients in human subjects using randomized placebo control designs. So based on our experience, when tested under rigorous human control conditions, the benefits are often more modest, highly variable, or dependent on individual factors like baseline metabolism and gut microbiota. We will discuss these factors later in this lecture. Here on the right side, you can see some of the clinical trials that we performed previously, and we have published these studies. But due to misleading or overhyped information from preclinical studies, from in vitro or in vivo studies, we normally do not pick food by active ingredient for research from the literature. We always do preclinical analysis by ourselves, and then we perform clinical trial based on our results. Because in our food-based human intervention studies, we observed that the effects on glycemic control, lipid profile, and even inflammatory markers were not consistent across all participants. Even if we test same food supplements for same population, we can see variable response of participant to that food supplement. Importantly, these outcomes dependent strongly on individual baseline metabolic status and their gut microbiota composition, which means that same dietary intervention may benefit some individuals while showing a limited or no effect in others. So in short, we can also say that popular claims often oversimplify biology because human evidence shows complexity in effects of these food supplement ingredients. For the claims, I have mentioned some of these like glycemic control, lipid profile, GIT issues like functional constipation. But one of the major issue is also in cosmetics. People are using different nutraceutical products for cosmetic use, but they do not observe same effects like it claims. So why many food supplements fail clinically? If I read the literature, or if I read claims about some food by active ingredients, there they mention that this food supplement is effective for glycemic control. This is effective for acne and or for functional constipation. But when we use that compound in human trials, they disappoint us. Why? The three main reasons that we observe in our research. First is poor standardization of active compounds. Unlike pharmaceuticals, nutraceuticals are not always standardized to contain same amount of active ingredients. For example, aloe vera contain some of polyphenols, but when we use it as part of food supplement, so it may not contain the same amount of polyphenols that was previously tested for preclinical studies. The second one and most important one is unknown or low bioavailability of the compound present. If I am testing food supplement or I am taking food supplement and I observe that the bioavailability of food supplement is quite significant, but if the bioavailability of that active ingredient, which is basically responsible
Dr. Hamad, you are inaudible, sir. आपका mic mute हो चुका है.
it is okay now?
Yes, sir, it's working now.
Okay. Here I am sharing example of the clinical trial that we tested a food supplement named Rusoporo that contains monocolines. This food supplement basically contains the extract from red yeast rice, and red yeast rice is basically designed as a functional food to contain monocolines, gamma oryzanol, and gamma aminobutyric acid. Monocolines act same like statins. It is effective against blood lipid levels. But here we designed a range of clinical endpoints. We included the lipid endpoints. We also included the glucose and insulin. We also included body weight and lipid profile. But what results showed us? It improved blood lipid profile, but had no effect on other parameters like blood glucose and insulin levels. So from here, we got a point that inappropriate clinical endpoints can also render the food supplements as ineffective in specific conditions. Let me share 3 examples from our human studies, including research, design, and also the endpoints. And I will show you that how we designed the study and what results we got. The first one is about Rusoporo that we discussed in last slide. Rusoporo was tested in mild dyslipidemia. Participants were enrolled that were with mild dyslipidemia. It was basically a placebo-controlled study, and participants were supposed to take the food supplement for 3 months. In the results, you can see that it significantly improved the lipid profile, but had no effect on blood glucose level and other parameters. Here you can see that it significantly reduced total cholesterol and LDL cholesterol while improving HDL cholesterol. Interestingly, it showed the effect on triglyceride was not significant, but in the literature, we observed that this food supplement had a significant effect on triglycerides, but those evidence were from observational studies, not from placebo-controlled studies, and also from the animal studies. So in human studies, we observed that the effect on triglycerides were not significant. In the second clinical trial, we designed a food supplement named JX Plus that contained dietary fiber-rich bioactive ingredient. The food supplement was tested in adults with mildly impaired glucose tolerance, and the outcome were postprandial glucose and insulin responses. Before moving into clinical study, we characterized the food supplement for dietary fibers, and you can see that it contained increased amount of resistant starch, ribinosylanes, beta-glucans, and ferulic acid. And here you can see the results. After 120 days, it showed significant improvement of blood glucose and blood insulin levels as compared to placebo. Basically, this food supplement, as it contained dietary fibers, it slows the absorption of postprandial carbohydrates. So that's why it led to significant reduction of postprandial glycemia and also improving insulin levels. In the third clinical trial, we picked Malva sylvestris. We made a food supplement. We chemically characterized Malva sylvestris before moving into clinical trial. The food supplement was tested in adults with functional constipation. This was basically a survey-based study because we picked Malva sylvestris from the literature and we did not know that whether it will be effective or not. So first we designed a monocomponent food supplement. We tested in adults with functional constipation. The primary outcome was bowel movements per week, while the secondary outcome was stool consistency and abdominal pain. And here you can see the results that after 10 days, it showed improvement in bowel movements, in Bristol score, mean stool consistency, and also abdominal pain. And the results were consistent while the participants were taking the supplements through 20 days. Here is the results of the chemical characterization. The food supplement contained a significant amount of polyphenols and also insoluble fibers. Besides, it also contained lipids, carotenoids that are also effective in some sort of constipation mechanisms. So now let's come to the point. Why do some people respond better than others? If all of us are taking the same food supplement, so it doesn't mean that everyone will respond same. There are some factors that lead to variable response of individuals to the same food supplement. The most important is gut microbiota.
Sorry to interrupt here.
Yes.
Sir, your voice break हो रही है.
अभी आ रही है?
Yes, sir, अभी आ रही है.
Okay, so the most important factor is the composition of gut microbiota. For the food by active ingredients, the most important interaction is with the gut microbiota. Most of the mechanisms that food supplement produce clinical effects is its potential to modulate gut microbiota, and in return, the potential of gut microbiota to metabolize the food by active ingredients. But not all individuals contain the same amount and same composition of gut microbiota strains. So it may lead to inter-individual variability of the response. The other factors are metabolic status. Everybody do not have same metabolism. Some are fast metabolizers, some are slow metabolizers. And as I just told you that the food by active ingredients do not produce effect by itself, but its metabolites do. So if a person, if two persons have different level of metabolism, one is fast metabolizer, one is slow metabolizer, the both individuals have a difference in gut microbiota composition and abundance of gut microbiota. So it is for sure that they will produce different response to the food supplement. One will produce improved response and one will not produce any significant response to the food supplement. The third and most important one is dietary and lifestyle contexts that may modify the outcomes. It may also include fiber intake. Fiber intake improve gut microbiota composition. So if we improve gut microbiota composition with the fiber intake, it will automatically improve our response to the food supplement. Similarly, the diet very rich in carbohydrates except dietary fibers and very rich in lipids. It also negatively impact gut microbiota. So it may also negatively impact our response to the food supplement. Here I am showing you a clinical study that we performed. In this study, we tested the effect of propolis that was rich in polyphenols in healthy and disease subject. Some individuals, some groups were healthy, but also the other groups had inflammatory bowel disease, and some had also a problem with obesity. And we tested the interaction of propolis with human gut microbiota in both of these individuals, that whether propolis impact gut microbiota similar in healthy and disease subjects. But what we got the results. Healthy individuals showed modest or no microbiota shifts while taking propolis. But on contrary, diseased individual exhibit pronounced microbiota modulation, and that led to improve their overall health. Metabolite responses were stronger in metabolically compromised subject. Metabolite response mean the formation of gut microbiota metabolite that are short chain fatty acid. The functional effects were minimal in healthy but evident in diseased groups. So if I am healthy and I am taking propolis for obesity, so it is of no use for me. If I carry a risk to obesity, so it means that I have disturbed gut microbiota abundance and composition. So in that case, if I am taking propolis, so I will produce effect. So now let's come to the point. How clinicians and consumers should interpret food supplement. Before this, I was talking more about research. So it is not easy to predict responses based on research, even clinical research. It is important to have some knowledge of research study design of clinical trials. So then we can predict responses of food supplement or even pharmaceuticals. So based on our trial experience in clinical trial, the most common misinterpretation is assuming that any statistically positive study automatically translates into real world benefit. And we already faced it. We sometimes picked food bioactive ingredient from the literature based on preclinical studies or observational studies. But in clinical trial, it showed modest or no effect. In other words, I can say that clinically tested does not always mean clinically meaningful, because there could be some problem. The duration of the study is short. So what if a food supplement is tested for 1 month in 50 subjects? It shows significant effect. But if I am using that food supplement for 6 months and in 1,000 subjects, so will it produce same effect? It is not always. The effect will automatically go down when we increase the duration of the study, or when we increase the sample size. So if I am performing a clinical trial, it is important for me to have a solid study design with significant number of population in the the sample size, and also increased duration of the study. For instance, normally for studies involving glycemia and lipidemia, we do clinical trial minimum for 3 months, but normally up to 6 months. Up to 3 months, it do not show a pronounced effect. But if I am testing it for 6 months and I get results that is clinically meaningful, so I can say that yes, these results are clinically meaningful in real sense, because with me the sample size were large, and also the duration of the study was sound. Similar is the case for inflammation. We tested a food supplement before for fatigue. we tested the food supplement for 3 months, and it showed significant effect against the fatigue, but the inflammatory parameters that are associated with chronic fatigue, our food supplement did not show any significant effect on those inflammatory parameters, because we tested the food supplement for 3 months, but for inflammatory mediators associated with chronic conditions, we must have study duration for at least 6 months. So in another trial, we increased the study duration to 6 months, and then we observed the results. It showed significant effects on inflammatory parameters too. So what truly matters? Duration of the study, appropriate endpoints, and standardization of the bioactive components. With the standardization, bioaccessibility and bioavailability are also important, not only standardization. If I am testing a food supplement and it contained curcumin 50 mg, but curcumin is normally low bioavailable. The bioavailability of curcumin is below 10%. So if I am testing the food supplement based on curcumin in clinical trial, it will not show me any good effects unless until if I increase bioavailability of curcumin through different parameters. So in short, clinically tested does not always mean clinically meaningful. Food supplements should always be interpreted through the quality of evidence, not the strength of claim. Not like that I gone through 10 clinical trials that showed that curcumin is effective for liver enzymes. Curcumin is already low bioavailable. I will need the quality of evidence. I will see that what about the bioavailability of curcumin, what about the duration of the study, and what about the endpoints. So looking forward, the field of food supplement is evolving to improve the effectiveness of these products in general public. The field is heading towards precision nutrition, where the supplements are designed to target specific metabolic and microbiota profiles. So in precision nutrition, we design the food supplement based on metabolic and microbiota profiles. Microbiota informed products, it is similar to precision nutrition. And tightening of regulations. Because we have stronger demands for human evidence. So standardization should and everything regarding regulatory like efficacy and safety parameters, they always be transparent. In short, the future belongs to food supplements that are clinically validated, not just commercially attractive. It is not just like I go to pharmacy and I see that there are so many nutraceutical products on the shelf. There is zinc, there is vitamin D containing food supplement. And I am taking different food supplements for vitamin E for my hairs, zinc for reversing aging, curcumin for improving my hepatic health. But there are some critical issues with these supplements. Before we need to address those. In developing countries, even in Pakistan, we have major issue that most of the nutraceuticals are not standardized and they do not contain the actual amount of the bioactive compound that is labeled on that supplement. So this is also one of the major problems that we are facing in our country, and how we can address this by tightening the regulatory affairs. Thank you very much. If there is any question, so you can ask me. Again, accept my apologies if there was problem with the pitch or with the sound. As I told you before, I am traveling, so maybe the connectivity was not that much sound.
Oh no, sir, that's perfectly all right. We are very thankful to you for the session. So, attendees, today's session reminded us that the real impact lies beyond the clinical and preclinical studies as well as the literature findings. It lies within the clinical rigor because clinical efficacy is in the human biology is dependent on the multifactorial nature of the supplements and their responses. Now we are moving on towards the question answer session. If you have any questions, you can drop them in the chat box or you can raise your hands and we will take the questions accordingly. Thank you very much.